Somewhere beneath a rotting log in Germany, a creeping harvestman is doing something that almost no other creature in the forest can. He is actively hunting snails. Not eating a snail that has died or has been crushed by a passing boot, actually tracking a living snail through the leaf litter and systematically tearing its shell apart with a pair of pincers so large they extend past the front of the body like a pair of grotesque arms. The creature is Ischyropsalis hellwigii. It is an opiliones, a relative of spiders and scorpions, and it has spent the last several million years becoming extraordinarily good at this specific thing. Harvestmen are familiar to most people as the pale, spindly daddy longlegs that clusters on autumn walls and skitters through gardens during the summer evening. But that familiar creature belongs to a completely different part of the harvestman family tree called the eupnoi. Ischyropsalis hellwigii belongs to a rarer group called the dyspnoi, which is limited entirely to the northern hemisphere and boasts a meager 330 species worldwide. The Ischyropsalis genus is limited to Europe, and only small bands of Europe at that. Only 24 species are known and are limited to living in cool, damp, hidden places. Within the dyspnoi, I. hellwigii belongs to the family Ischyropsalidae, a group morphologically extreme and ecologically distinctive. The family historically contains only a single genus, Ischyropsalis, with 24 described species, all restricted to Europe. Currently, the family contains an additional 2 minor genera, only found in North America. The genus name Ischyropsalis means strong shears, as in scissors. What defines every member of this genus, and what makes them instantly recognizable, are the chelicerae, a pair of appendages near the mouth, functionally the jaws, that have been enlarged to excess. In most arachnids, the chelicerae are small, pinching appendages used to manipulate food. In spiders, they carry the venom glands and are used to inject prey. In I. hellwigii, they become 2 massive, 3-segmented arms, each composed of a thick basal segment and 2 articulated claw-like distal segments that together can equal or exceed the length of the animal's entire body. So what on earth does it do with these things? The answer, confirmed by captive feeding experiments and field observations, is that I. hellwigii uses its giant chelicerae to crack open the shells of living land snails and eat them. This makes it one of the very few arachnids on Earth that has evolved a dietary specialization on mollusks, and almost certainly the only harvestman known to do so as a primary feeding strategy. The behavior was first systematically documented by the German arachnologist Jochen Martens in 1965 in a series of captive experiments that have since become the foundational reference for the species' natural history. When I. hellwigii finds a snail, the animal approaches the gastropod and begins to work at the shell opening with its chelicerae, inserting the tips between the shell lip and the soft body of the snail. Rather than attempting to enter the shell through its natural aperture, I. hellwigii instead breaks the shell itself, chipping and levering away fragments until enough of the shell wall has been removed to access the body. The shells left behind by this process are distinctive, irregular broken out fragments with large and less symmetrical damage patterns than those left by other shell-crushing predators. In captive experiments, I. hellwigii consistently accepted snails of multiple families when offered, suggesting the preference is for gastropods broadly rather than any specific shell architecture. When insects were offered in the same experiments, the harvestmen refused to eat them. This rejection of insects is a striking result. Most arachnids, and most harvestmen broadly, are generalist predators that will take almost any accessible soft-bodied prey, and deliberately declining an insect in favor of a creature armored in calcium is not an obvious dietary choice. It implies that the dietary preference for snails is deep-seated and specific, not simply an artifact of what the animal encountered first, or most often in its environment, but an evolved preference. This stands in contrast to other members of the genus who readily feed on other invertebrates. The mechanical demands of shell cracking shed light on a fascinating anatomical comparison. A 2024 study reconstructed the shape of I. hellwigii's chelicerae and modeled their range of motion. The study compared I. hellwigii to its close relative, I. muellneri, a cave-dwelling species with a different diet. The results showed that I. hellwigii's chelicerae, despite appearing larger and more imposing, actually have a smaller range of angular motion than those of its cousin. The joints are stiffer and construction more robust. This is the signature of a crushing weapon, optimized for force over flexibility, like a pair of pliers built for maximum grip rather than reach. By contrast, I. muellneri, which lives in caves and feeds on whatever slow-moving prey it can find, has longer, thinner chelicerae with greater flexibility in all joints. A cave predator facing food scarcity needs versatility. I. hellwigii, a surface specialist with a reliable and abundant prey base, has traded that flexibility for the raw mechanical power needed to crush shells. Beyond the chelicerae, I. hellwigii presents the general appearance characteristics of the dyspnoi, a compact oval body with moderately long legs and colored in dark blackish browns that blend in well with the dark forest soil and rotting wood. Like most harvestmen, the eyes sit on a small raised turret near the center of the body, but its vision is terrible and has no ability to form detailed images. It navigates its world primarily by touch and by contact chemoreception. In most harvestmen, the second pair of legs serve as a specialized sensory organ, sweeping the air ahead of the animal during movement and carrying dense arrays of chemoreceptors. In I. hellwigii, this function is somewhat redistributed. The enormous chelicerae dominate the front of the animal, and the sensory legs must navigate around them. The palps, the pair of smaller legs beside the chelicerae, are long and slender, used for touching and probing the environment rather than for seizing prey. The body surface of the harvestman is largely smooth and hardened into a tough exoskeleton, which is typical of the dyspnoi, and provides both structural integrity and some protection from physical threats. Ischyropsalis hellwigii is the most broadly distributed species in its genus, and the one most likely to be encountered. Its range extends across a broad swath of Europe, from northwestern Germany and the Netherlands, southward through Austria, the Czech Republic, Slovakia, Poland, and into the Balkans. A recent study recorded a subspecies west of the Rhine in the Eifel region of Germany for the first time, confirming that populations continue to be discovered in areas where the species was previously unrecorded. The full extent of the range, particularly towards the east and south, remains incomplete. Habitat preferences are consistent across the species range. I. hellwigii is a creature of cool, humid, mature forests. It is found most reliably in old-growth or semi-natural deciduous woodland, particularly with beech trees. Within these forests, it occupies the soil surface and the zone immediately beneath bark, stones, and decaying logs, where the temperature and humidity are buffered, and the leaf litter is deep enough to harbor abundant amounts of snails. The species, like many harvestmen, need high humidity for activity, and is most mobile when forest floor moisture is at its seasonal maximum. For I. hellwigii, summer is essentially the low season. The forest floor is drier, the animal is less mobile, and activity concentrates in microhabitats, beneath logs, in the immediate substrate of stream margins, or in the deep layers of undisturbed litter. Juveniles are thought to pass through the summer in concealment beneath bark and in deep litter, growing slowly in conditions limited by food, before entering the autumn season as newly matured adults. The complete developmental period, from egg to adult, is believed to take 2 years, making this a relatively long-lived harvestman. Perhaps the most biologically unexpected feature of I. hellwigii is what happens when two individuals meet for mating. In many animals, courtship is a matter of visual display, acoustic signaling, or pheromone signaling from a distance. I. hellwigii is nearly blind, living in the dark beneath forest logs, and does none of these things. Instead, when a male encounters a receptive female, he offers her a taste of a secretion produced by a specialized gland built into the base of his chelicerae. The gland in question sits in the thick basal segment of the male chelicera. During courtship, the male presents his chelicerae to the female, and she appears to drink this secretion before mating. This gustatory gift is the signal that determines whether mating occurs, and its chemical properties are specific to each species. The chelicerae of I. hellwigii are simultaneously a shell-crushing weapon capable of breaking calcium, a sensory probe used to explore the environment, a delivery platform for species-specific mating signals, and, in females, a dexterous prey handling tool. That a single appendage serves 4 distinct functions across 2 sexes is extraordinary evolutionary repurposing. It also means that the cheliceral morphology of male I. hellwigii has been shaped by at least 2 competing selective pressures: the mechanical demands of snail predation and the social demands of mating. For a species described in 1793, I. hellwigii remains incompletely understood in ways that should give anyone pause.